I'd like to start by acknowledging the Dharawal, Yuin, and Wadi peoples as the traditional custodians of country in the Illawarra. We'd like to pay our respects to elders past, present, and emerging, and to all Indigenous people living within the region, and extend that respect to other Indigenous people with whom we engage. Through our research and engagement, we seek to help others appreciate and act in respect of the significance of country. I'm David Safadi. I'm a PhD student at the University of Wollongong with the David Adams Lab, and I'd just like to remind the audience to please keep on mute while talking and save questions for the end if possible. Today we're joined by Renae Ryan. Renae is a professor of biomedical pharmacology at the University of Sydney. She received her PhD from the University of Sydney in 2004 and completed postdoctoral work at Columbia University and the National Institutes of Health. Renae then returned to the University of Sydney in 2007, where she leads a research team that investigates the molecular mechanisms of neurotransmitter and amino acid transporters and their role in neurological diseases such as episodic ataxia and cancer. She has received several awards for scientific excellence, mentoring, and outreach, including the 2023 Nancy Mills Medal from the Australian Academy of Science and a New South Wales Tall Poppy Award in 2011. She has also just received a Member of the Order of Australia for her significant service to science as a researcher and her contributions to diversity and inclusion. Renae is a globally respected leader and advocate for gender equity, diversity, and inclusion, and a sought-after supervisor, mentor, and role model for women in science. Her talk today will be looking at the twisted link between a dual-function glutamate transporter and episodic ataxia. Excitatory amino acid transporters regulate excitatory neurotransmission through transport of glutamate into cells, mostly glia, to terminate neurotransmission and avoid neurotoxicity. The molecular mechanisms that allow these transporters to carry out these roles, including the conduction of chloride ions, are currently not clear. So today, Renae will be presenting a cryo-electron microscopy structure of a glutamate transporter homologue in its open channel state, revealing an aqueous accessible chloride ion permeation pathway that is formed during the transport cycle, and discusses the impact of a series of mutations in EAAT1 that have been identified in patients with neurological disease episodic ataxia type 6. By studying EAAT function, and using a Drosophila melanogaster model of locomotor behaviour, the results indicate that mutations that lead to functional glutamate transport, but are either increased or decreased in chloride ion channel activity, contribute to the pathology of episodic ataxia, highlighting the importance of chloride ion homeostasis in glial cells for proper central nervous system function. Her findings provide insight into the mechanism by which glutamate transporters support their dual functions and provide a framework for the rational development of therapeutics that can differentially modulate substrate transport or channel properties for the treatment of neurological disorders caused by EAAT dysfunction. So looking forward to that, and whenever you're ready to start sharing your slides, and you can take it away.
Okay, thank you, David, for that thorough introduction. You've nearly done my talk for me, but hopefully I'll still be able to show you some exciting results today. And I'd just like to acknowledge that I'm in the lands of the Dharawal people. I'm actually at the Westmead campus of the University of Sydney today. So thank you very much for the invitation and, as I said, for that kind introduction. So I will just share my screen. Hopefully you can see that. I might just put my laser pointer on too. Okay, I'm going to get rid of these photos and hide that as well. Right. Okay. There we go. So as David mentioned in his introduction, Our group studies glutamate transporters, which are also known as EAATs, or excitatory amino acid transporters. And that's because these transporters, in addition to glutamate, can also transport another excitatory amino acid, aspartate. They're found throughout the body and in many different organs, and really play a role of clearing glutamate and aspartate into cells. But in the brain, they've been most widely studied for their role in synaptic transmission. So here's just a cartoon of a glutamatergic synapse. The little red dots are glutamate. They're packaged into presynaptic vesicles, and upon an action potential reaching the end of this neuron, these vesicles will fuse with the membrane and glutamate is released into the synaptic cleft. It will traverse the synaptic cleft and activate glutamatergic receptors to do its job for chemical communication in the brain. But for these neurons to transmit information efficiently, the glutamate really needs to be removed from the cleft rapidly, and it has to be controlled in a way that it doesn't spill over to other synapses and activate them. And this role is really carried out by the EAATs. There are 5 subtypes. EAT 1 to 4 is shown in this diagram. EAT 5 is a bit more localized, specifically localized to the retina. But EAT 1 and 2 are found on glial cells. As David said, they really clear the bulk of glutamate from synapses and are very highly expressed on the surface of glial cells. EAT 3 and 4 are found on the postsynaptic neuron and are thought to prevent spillover of glutamate into other synapses. So the glutamate transport is coupled to the co-transport of other ions, and that's because you have to push glutamate against a very large concentration gradient. So there's about 1 millimolar of glutamate inside cells, and the resting concentration of glutamate is thought to be as low as 25 nanomolar in the extracellular space in the brain. And this, the power to drive glutamate is really derived from the coupling of 3 sodium ions and 1 proton moving into the cell, and this is followed by the counter-transport of 1 potassium ion. The dysfunctional downregulation of glutamate transporters has been linked to pretty much any neurological disease you can think of, because of course, uncontrolled glutamate concentrations in the brain will eventually cause excitotoxicity. And they've been linked in both acute and chronic conditions. So acute, for example, ischemic brain damage following a stroke, but also these longer-term neurodegenerative diseases. In addition to the coupled glutamate transport, which has been very widely studied, the binding of glutamate and sodium also activates what is called an uncoupled chloride conductance through these proteins. And so this means it's uncoupled in that it's not stoichiometrically coupled to the transport process. Once the chloride channel is open, chloride will just flow down its concentration gradient, but it does require glutamate and sodium to be bound to the transporter to be activated. And we've been really interested in studying this chloride channel function of the glutamate transporters for a long time. I actually started this in my PhD back with Renae in the early 2000s, and he was actually involved with the group that discovered the existence of this chloride channel. And it's interesting to us because it seems to be found in all members of the family studied thus far, including some bacterial and archaeal homologues. And we don't really know what it does. Some of the roles proposed for the physiological function have been charge neutralization, because for every glutamate molecule moved in through the coupled process, you get an inward movement of 2 positive charges. And so it's been proposed that maybe having this anion leak can counterbalance that and allow the transporters to function over a wider range of conditions. It could also be involved in homeostasis and osmotic balance as you're moving ions and water across the membrane through these proteins. In the retina, EAT 5, it has actually been shown to have a clear physiological role, and these transporters are actually expressed on the presynaptic terminal. And when they're activated, they have a very large chloride conductance, and they're very poor transporters actually. So they kind of act like glutamate-gated chloride channels, and that leads to a negative feedback loop, which reduces further glutamate release. But that's really the only clear kind of physiological role that's been proposed thus far. We started getting really interested in this as well, not just from a mechanistic point of view, which I'll touch on in the next slide, but also because the chloride channel function started being linked to a disease called episodic ataxia type 6. And I'll spend the second half of the talk talking about that disease. So really, from a mechanistic point of view, it was interesting to understand how a protein could act as a channel and a transporter. Because, of course, if you remember your undergraduate days, we know that channels are a continuous pore that is open where ions will flow down a concentration gradient. These processes are generally very fast, different ion channels and receptors, ligand-gated receptors that have this function. Transporters, on the other hand, and particularly secondary active transporters, which are pushing a substrate against a concentration gradient, can never be open to the same side of the membrane at the same time because they'll just allow glutamate to flow out of the cell. So to be able to move glutamate into the cell, there has to be this controlled alternating access mechanism. So how does one protein support these dual functions of both transport and channel activity? And we have used, as many others have for many years now, an archaeal homologue of the glutamate transporters from an organism called Pyrococcus horikoshi, and this protein is called GLPPH, and it's been a very good model for the glutamate transporter family, and it has very high amino acid identity to the human transporters. And so we know from many structures that these proteins exist as trimers, and here we're looking at the trimer from the top outside of the cell, kind of top down in both a ribbon diagram and space filling diagram. And here we're looking in the plane of the membrane. And so three identical protomers come together, and they are held together by this brown portion, which we call the scaffold domain or the trimerization domain. So that's the part of the protein that interfaces with the other protomers. And it also has this blue domain, which is we now call the transport domain. And this transport domain contains the binding sites for substrate and ions, and also undergoes quite significant conformational changes, which I'll show you in a moment. And so each of these individual monomers that make up the trimer are identical. So we generally just show the individual monomer when we're talking about the protein, but remember it is a trimer, and there are three copies of this working together. So GLPPH, there's been many structures solved, and this was really the first transporter that was identified to use an elevator mechanism of transport. So of course, for many years, the alternating access mechanism of transport was what everyone transporters used, and that MFS fold is very much like that, open to the outside, closed, open to the inside with this centralized binding site. But what we now know from GLPPH and all glutamate transporters is that they use this elevator mechanism, and I'll try and explain it to you here. So here we have an empty transporter facing the outside of the cell, and if you focus on this red hairpin loop, which is like a gate, you can see it's open here. Then we have substrate and ions come in, and then the gate closes, and then you can see this transport domain in blue with the red gate moves all the way down, and then the red gate opens again and releases substrate and ions into the cell. So this is what we call the elevator mechanism. I'm hoping you can see my hands. I can't see you, but you have the open gate, it closes, and then this transport domain moves down and actually rotates as it moves down into the inward open and releases in the other side. And you can look at this in space filling representation as well. And what you can see, or what we observed from all of these structures, and there's been many structures, was that in none of them you couldn't see a continuous aqueous pathway. There was nowhere that water and ions could be getting through in a channel-like fashion. Except one intermediate structure that was solved by my colleague Volker Budka started to show that there was an aqueous pathway that was starting to open up between the scaffold and transport domains in this intermediate structure. And we were really interested in this because a lot of the residues we had studied over many years that specifically impacted the chloride channel component but did not impact glutamate transport function were found to line this cavity here. And so the hypothesis of the study that I'm going to tell you about in the first half of the talk was really that the chloride channel we hypothesized the chloride channel would form as that transport domain moved down further. So somewhere in between this intermediate state and this inward facing occluded state, we would get a continuous pathway at this interface. But it proved very difficult to show this. And we know from all of these structures that the transporter really likes to be in this, these outward occluded states and the inward occluded states. The intermediate state was actually an accident that was trapped in a disulfide cross-linking experiment that kind of went a bit awry. That the transporter was really stable in those two end states. So we used a disulfide cross-linking strategy to try and trap the transporter between this intermediate state and the inward-facing state, because this is where we thought we would see the chloride channel open. And this work was done initially started by Rosie Cater and followed on by Ita Chen and Pep Font, who did a lot of the structural biology, and Quinny Wu as well, who did a lot of the functional electrophysiology experiments I'm going to talk about today. So what we did with this cross-linking strategy was introduce one cysteine into the static scaffold domain and one cysteine into the transport domain. And we did about 30 of these different pairs to try and trap the transporter as it moves through its cycle. So the great thing about this is we can obviously use structural biology to study GLTPH, but we can also study the function of EAT1 using electrophysiology because these transporters are electrogenic. And one thing just to note about the electrophysiology, and I'm not going to go into huge amounts of detail, but the total conductance we see is always a combination of both of these functions. So the glutamate transport function and the chloride channel function. And if you could look at them alone, the glutamate transport would never actually cross the x-axis, so it would never reverse direction. And the chloride conductance alone would have a very negative reverse potential around -20, and it would have this kind of linear current voltage relationship. And so we can look at the crosslinks functionally to see if we are indeed trapping the transporter in a chloride conducting state, where we would hope to see a conductance that look more like the red line. So as I said, we made about 30 of these different crosslinks, and I'll just show you a couple of examples here. This is one, and all of them actually, most of them cross-linked in some way, but many of the crosslinks we saw just inhibited both transport and chloride. So here we can see in black, it's a bit hard to see under the orange, is the control. If we then treat the oocytes with a reducing agent DTT, we see an increase in the current. And that suggests that there was some kind of native crosslink between these two residues, which were trapped in the outward facing state. But then when we incubate the cells with the oxidizing agent copper phenanthroline, which should reform the disulfide bond, we saw that the current went back. So this was great. It showed that we were getting cross-linking between these residues, but we were inhibiting both of the functions at the same time because there was no change in the reverse potential. So we were inhibiting both chloride and glutamate at the same time. And as I said, many of them were trapping in the outward-facing state or the inward-facing state, and that makes sense. We know there's no chloride channel in those structures. But we did find one in particular that did exactly what we wanted it to do. And so this double mutant we saw had very negative reverse potentials and these linear conductances, which suggested that we were actually trapping this transporter in a chloride conducting state. We solved many structures with crystallography at the time. That's what we were using. This was project had been going on for quite a while, but this double mutant here, we could never really, we got crystals, but they would never really trapped below about 6 angstroms. And so we got very frustrated trying to work with this. Pep and Icha tried a lot of different things, different conditions, and we just could never get we could not improve the resolution. So we turned to cryo-EM, which was becoming much more accessible and a much more useful technique for membrane proteins. And we did this work in collaboration with Alastair Stewart and Megda Sobti at UNSW. And what we did was looking at that double cysteine mutant, we collected images, went through the process and got some really nice 2D models. And you can even see the threefold symmetry on these. You can imagine you're looking down the top of the trimer there, and you can see those 3 white dots, which we think might be hairpin 2, and got some really nice 3D models and structures out of the data. This was actually collected on Antarctica at UNSW. But we were quite disappointed because the structure we ended up getting out was, lowish resolution around 3.9, which is not that dissimilar to actually a lot of the crystal structures of this protein. But it was in this intermediate outward facing state, which we'd already seen in crystallography, and it didn't have a continuous pore. But what we then did with Alastair and Megna is actually use symmetry expansion, because the beauty of this trimer is that it does have threefold symmetry. And so we pulled out each individual protomer from all of those trimers and reclassified them. And again, we got about 9 classes that were in this intermediate outward facing state, but I wouldn't be telling you the story if we didn't get something different, which was one group, about 15% of all the protomers was in this chloride conducting state. And so this chloride conducting state was indeed in between the intermediate outward facing state and the inward facing state. And what we're showing here is a slice through of the protein in all of these different states. And this is showing this partial cavity that was opening up in the intermediate state And you can now see that there's a continuous cavity that's going right through in between scaffold and transport domain. And this was worked out really nicely in that this serine 65 residue we knew was really important for determining the anion selectivity of this, or the permeability sequence. And in this structure, it was still very closed around this region. But in the chloride conducting state, it's opened up right up in this region. And now the narrowest region of the pore is around this arginine residue, which Rosie had actually shown determines that this actually is selective for anions over cations. So it made nice sense with previous mutagenesis data. So why did we fail miserably with crystallography but succeed with cryo-EM? And it's because we actually had a non-homogeneous population. What we had was this chloride conducting state, but we still had the intermediate outward facing state. But in both states, we did see cross-linking. It's just that in this state, the blue residue here is above the red one, and in this state, it's below the red one. And so the cross-linking strategy worked, but I suppose we didn't anticipate that we might be able to trap two different states with the same cysteine pair. And you can see here that the cysteines are in cross-linking distance in both of these structures. And one thing that the reviewers actually asked us to do, which was a bit of a pain at the time, but I think was quite nice in the end, was reconstruct the trimers to try and get an idea about whether you could have combinations of intermediate and chloride conducting in a trimer. And indeed, we did see that. So again, by far, the cyan is the intermediate state. Most of the protomers were in this state, and we could have all intermediate, all chloride conducting, and then a combination of the two. So it was a pretty rare state that we could see. With this, it was less than 1% where all three were in the chloride conducting state, but it did allow us to solve the structure of this protein in a channel state. So once we had that, we then turned to our collaborators, Shashank and Ahmad, to do some MD simulations, because of course we wanted to see that water could indeed, and chloride ions could indeed permeate this pathway at the interface of the two domains, which they saw quite easily. And then what Shashank did was he could identify every residue that was interacting with chloride as it was moving through the pathway. We also looked at EAT1 to confirm that this was the case, because of course our functional data was in EAT1, but our structures were in gel to pH. And here you can see a chloride ion in purple as it's moving through the interface of the transport and scaffold domains. And Shashank could monitor the interactions with every residue and see that there was a couple of regions that were a bit more difficult for the chloride ion to get through, and then a region which was more favourable. And this aligns with this arginine residue I talked about before. And so we described how we described these hydrophobic regions, where they're two hydrophobic gating regions in the chloride conduction pathway that we referred to as an intracellular gate and an extracellular gate. And so these were regions that were made up of hydrophobic, where the chloride ion could get through, but it was an energy barrier to pass through. And so then what Quinny did was look at these residues in EAT1 and just mutate them all to alanine. So just got rid of the hydrophobic side chains to an alanine. And what she found was that when she did remove the kind of hydrophobic bulk of these residues in both this intracellular and extracellular gate, the chloride ions move through more easily. And this is represented by this negative shift in the reverse potential. So removing hydrophobic bulk increased the chloride movement, or the ease of chloride movement through the pathway, which was nice because it agreed that this was indeed the pathway and the chloride ions did have to get through these two gating regions. One of the residues, alanine 289, was already an alanine, and so for fun, Quinny decided to introduce a phenylalanine at this point. And this was completely the opposite effect. It shifted the reverse potential to as far right as we can actually measure in our system, really preventing virtually any chloride movement going through. So when there's a phenylalanine pointing into this region, it's really blocking the chloride ions. And I must point out, I don't have the data here to show you, but you have to trust me that none of these mutations affected the glutamate transport process. And that's what's really interesting about these transporters. We can specifically manipulate residues at that interface where the chloride channel pathway is, but we don't, that doesn't necessarily affect the movement of the transport domain and the ability to transport glutamate. Okay, sorry, I'm talking a bit fast because there's a lot to get through. So that first part of the talk, just wanted to show you that we have indeed identified this chloride conducting state, which is on the transport pathway in between the intermediate state and this inward facing state. A complete chloride pathway opens up at the interface of the two domains, and we've shown that in both GL's pH and EAT1. And this is really interesting because it as I said, it seems to be conserved in the family, and a lot of the residues that are both the gain residues and the residues that are selected for anions over cations are found in this pathway. And so what we're moving on to next, and Amy Cheng in the lab is working on this, is really trying to use this structure now to identify functionally selective inhibitors, because all the inhibitors we currently have transporter in this state up here, this open to the out state. And so you're inhibiting both the transport and chloride channel together. And what we'd love to do is to be able to have compounds that can selectively alter one function or the other. And this would be, of course, interesting to study the physiological role of the chloride channel in cells and animal models, but it's also may be useful therapeutically, because I'm now about to tell you about episodic ataxia and the link to this chloride channel function in EAT1. So episodic ataxia, there are many episodic ataxias. They're channelopathies and generally are found in, mutations are found in potassium and calcium channels. So EA1 and 2 are the most well-studied forms of episodic ataxia. And the disease is characterized by recurrent periods of ataxia and vertigo. So it's a little bit like epilepsy in that people can be quite normal and then have attacks, but instead of having a seizure, they have these periods of ataxia and vertigo and can lose all motor control of their bodies. As I said, EAT1 and EAT2, EA1 and EA2 are very well studied, but there is this EA6, which has been linked to the glial glutamate transporter EAT1. Now, there's only been a handful of mutations that have been studied and really clearly linked to this disease, to EAT1. And they're found throughout the protein. There's no hotspot for where these mutations exist. That's just showing a topology diagram, but you can see here on the structure, they're kind of all over the place. And so one residue that had been studied pretty well was this proline 290 to arginine mutation. This had been studied by a couple of different groups and shown that this mutation decreased glutamate transport. It had some effect on glutamate transport capacity, but it had a huge chloride channel conductance. And so there had been a proposal that potentially this disease was linked not just to glutamate transport problems, but also chloride channel problems as well. And a collaborator of ours, Don Ben Mail at McGill University, actually has developed a really cool Drosophila or fruit fly model to study this disease. And so here you have, these are both the mutation, but here you have the larvae in a normal state where you prod it and poke it with a wire and it will react and move and recoil from the touch. Here, it looks like this is dead, but it's not. This is the same mutation, but it's in a period of paralysis. And you can see that it's trying to move and it just can't. But right at the end of the video, hopefully you'll see it starts to move again. So these larvae are pretty unhappy, although they can crawl around, which is really important for them to feed and develop into flies. When they're having these periods of paralysis, they obviously can't eat. And these larvae don't really turn into flies. They die in the larval stage. And what's really cool about this fruit fly model is that they have their own glutamate transporter called DEAT1. You can knock out their glutamate transporter and put back in the human one, the wild type, and they completely recover. So we've used this model to now test all of these mutations in the fruit fly as well. So initially what Quinny did was make these mutations and study their glutamate transporter function. So here are just radio-labeled glutamate transporting to oocytes expressing these transcripts. And you can see compared to wild type here, most of them have some level of transport compared to wild type, except two, this P290R and another one, M128R, that had very, very little transport. And we already knew that about the P290R that had been established, but this M128R was also very, we couldn't see any uptake over background, and our background here are uninjected oocytes. So we can also look at the currents, as I showed you earlier, where we can apply glutamate to these cells and we see a nice current generated by the transport process, which you don't see in uninjected cells. And so Quinny just had a look at the substrate affinity for all of these proteins and also the sodium affinity, because again, in black, you can see wild type, and in red is this P290R that we'd already seen had a much higher substrate affinity, but a lower sodium affinity. So there was some ability of substrate and sodium to bind, but very poor transport rates. And you can see all of these other mutations were somewhere in between those, but they were all functional glutamate transporters, except for the M128R, where we saw absolutely no activity, and we've never really seen that with an EAT1 mutation before, and we've done many, many mutations. And so Quinny just wanted to check, is this M2R even expressed at the surface? And what she found when she tagged the transporters with GFP was, compared to uninjected oocytes down here where there's no fluorescence at all, there was fluorescence for all of the constructs. Clearly, these two were less, and this is just quantified here, about 30% compared to wild type. But we know that we can observe clear currents for this P290R. It's expressed, yes, lower compared to wild type, but it's definitely at the surface. But again, this M to R, we could see no glutamate activated conductance, even though there was some expressed at the surface. Quinny then wanted to look at the chloride channel function, because of course we knew this P to R had increased chloride, so a shift to the left in the reverse potential. It looked more like this over here. And what she found was quite interesting. A couple of these mutations really weren't very different from wild type at all. This threonine and cysteine mutation, and another couple seemed to shift the reverse or the other way compared to wild type, which is down here. And so these suggested that these had reduced chloride activity compared to the P290R, which threw a bit of a spanner in the works, because of course at the time our hypothesis, as other groups were working on, was it was this increased chloride channel component that was due to the link to the phenotype of episodic ataxia. But what we were seeing was a decrease as well. And just to touch on the M to R, because of course that's not on that graph because we couldn't observe any currents. Something that Quinny did notice, and I'll move through this fairly quickly, but it is what you can see in the absence of glutamate. If you do a voltage step with these cells, you actually observe these transient currents. So down here, if you just think, because I know this is most probably not an electrophysiology crowd, we normally hold our cells at minus 30 millivolts. And if you jump to a positive membrane potential, so this is just showing you the membrane step, you see these large transients and then a steady state conductance. In grey, light grey here is the uninjected cell. So you still see the transient, but then it comes back to baseline. But in wild type in the P290R, we see this leak conductance, which we know is due to chloride movement through the transporter. So this is in the absence of glutamate. And these transients are thought to be bumping sodium off the transporter. So when the transporters are just sitting there in the cell, they're generally bound to sodium because we have high sodium in our buffer. If you jump to a positive potential, you're just bumping off the positive sodium, but it will relax and then it'll rebind over here. So these are called transient conductances, and all you really need to take away from this is that in addition to the P290R, which we knew had a large leak, Quinny kept seeing these really large conductances for the M to R mutation. So this was really interesting because we knew it was at the membrane, similar levels to the P to R mutation, although to point out here that this is raw data and these are both around 30% of the level of wild type. So these have very large leak conductances. And what Quinny wanted to do was to figure out, well, what ions were carrying this conductance? We know that it's chloride in the case of P to R and wild type, and that's just shown here. When you change the chloride concentration, you see a big shift in the reverse potential, but that didn't seem to be the case for the M to R. That wasn't that different to uninjected cells. But then when she looked at all of the other ions it could be, it ended up being sodium. So when she changed the sodium concentration, she saw the biggest shift in reverse potential for this M to R. So it suggested that this leak was there, but it wasn't like the wild type leak, which was carried by chloride ions. It appeared to be carried by sodium ions, and that'll be important when I go on in a moment. So just to summarize here, all of the functional data showed really different things. So we saw that for P290R, we knew it had reduced transport, but it had increased chloride conductance and this increased leak. For these two, the valine and the alanine mutation, completely normal glutamate transporters. There was nothing wrong with their glutamate transporter properties, but they did have reduced chloride conductance. The M to R was very unhappy, but it appeared to have a sodium leak. And these two, in our hands at least, did not seem to have anything wrong with them at all, even though they had been picked up in EA6 patients and clinically had been linked to these mutations. So the methionine mutation was particularly interesting to us because, well, for two reasons. The first one was that it seemed to have the most severe effect in the patient. So it was picked up, or the patient started having episodes as a baby at 11 months old. It was picked up clinically or finally diagnosed. And the other thing with episodic ataxia is it can take a very long time to be diagnosed. When she was 10 years old, it was picked up and finally diagnosed. And although her development was normal, she had repeated episodes of ataxia, tremor, speech throughout her life, which are these periods of episodic ataxia. And so you can see that this child had the mutation, both parents did not carry it. And what was interesting for us as well was this methionine residue is highly conserved throughout the family. It's found in every glutamate transporter you care to look at. And of course, a methionine to an arginine mutation is quite a significant change in the amino acid residue itself. The other thing that was really interesting to us was that this methionine residue is very close to one of the sodium binding sites. So this sodium binding site, although it's called sodium 3, it's really important because it was identified last, but it's the sodium ion that needs to bind first for the whole transport cycle to work. And this methionine residue would be right here in between these two residues. So it would be facing out towards the lipid, but it's right near this sodium 3 site. So we wanted to investigate this a little more. We couldn't really use our functional experiments because we just couldn't get much out of it. And so we went back to our MD collaborators and asked them to have a look. And so this now is in an EAT1 model. And what you can see is that wild type has a methionine residue here, and this is with the arginine mutation. And here what Shashank did was he placed a sodium ion at what's called sodium 1, and you'll see in this simulation that this ion has to move down over here into the sodium 3 binding site, and it's coordinated by this aspartate residue 400. And so we know that this happens, and this is a really important step for the whole transport cycle to work. You can see it's moved down here. In the same kind of simulation period, and they even did longer, they could never see the sodium ion in this mutation moving down. It's not so surprising. You can see that there's an arginine here, an arginine side chain in place of the methionine, and it's actually pointing in and directly interacting with this D400, forming a salt bridge that therefore prevents the sodium ion coming down. So that would explain why we get this dead transporter, but it doesn't explain the sodium leak. And interestingly, when Shashank was looking at this with MD, he saw two conformations. So this he called the in conformation, where the arginine was pointing in, but he also saw a conformation where the arginine side chain was pointing out into the lipid membrane. And arginine residues are interesting in that when they do point out into the lipid bilayer, they're known to distort the membrane, and they can pull up the head groups of the and interact with the head groups of the lipid bilayer. And what Shashank noticed when this happened was that there was actually a continuous water wire or a pathway here from where the arginine pointing out into the lipid and forming an aqueous pathway into the cell. And so we think that this might explain the sodium leak situation, where you have a sodium ion that comes in. If the arginine's pointing out, it could move into that sodium 3 site, but it keeps leaking out because there's now an aqueous pathway for sodium ions to move into the cell. So either way, this transporter is extremely unhappy, but using a combination of techniques, we can see that we've been able to show that it clearly doesn't allow sodium 3 binding. If it does, and the arginine's pointing out into the lipid, it's most probably for this leak pathway. So very unhappy transporter. Not surprising that this girl has fairly severe episodic ataxia. Okay, so the other thing we can do, as I mentioned earlier, is in collaboration with Don Van Maal and all of these people are at McGill University, is we can look at the function of these mutations using the Drosophila assay. Instead of videoing the larvae, which is what they used to do in the old days, Tomoko and Alastair down here actually developed this worm, using worm tracking software, developed a Drosophila tracking software. And you can see that you can look at multiple larvae at once. And what the software does is it can track their distance, their movement, their speed, the arc of their movements, all of these different things which are useful to understand. And so as I pointed out earlier, first of all, you can see here wild type. This is just showing you the movement of a bunch of different larvae. Very quickly, you can see that there's some differences here between wild type and some of these mutations. So as I mentioned earlier, you can knock out the Drosophila, eat one, and you get very unhappy larvae. They can't move at all. And then you can put back in wild type human, eat one, and completely recover. So this is very important because it means that we can then look at the human mutations in this model. And so again, with the M to R, you can see here, they are not moving at all. So there's nothing periodic about this. They just do not move. It was actually hard to even pick up the larvae because they were so still. And this was the one, as I just explained to you, the very severe mutation. The V to I, there was very little movement, as was for the A to T, and both of these we saw surprisingly no effect on glutamate transport. So they're completely functional transporters, but they have reduced chloride. And then for these other two, the T and this cysteine mutation, we really saw nothing in our assays, and neither did Don. These seem to be just able to move and forage and develop as wild type. So at least in our hands, we don't think that these mutations are actually linked to the disease in these individuals, whereas the V and Allen, the valine and Allen mutation clearly are. But what was interesting about these mutations was as opposed to the proline mutation, which did have reduced glutamate transport and increased chloride, this really seemed to be a function of just having a reduced chloride conductance. And if we correlate the changes in the reverse potential with the changes in the ability of these larvae to move, interestingly, we get a correlation where we see the more severe mutations that impact chloride movement. So this is a reduction in chloride. Here's the A to V, and here's some other mutations that we've just studied in the past that really knock out the chloride channel. The more severe the chloride reduction, the more severe the phenotype seems to be in the larvae. And we can also look at this in the other way. These are a couple of mutations that increase the chloride channel very significantly, but don't affect transport. And we chose them instead of P290R because we already knew what that one did. We wanted to show that all of these have completely fine glutamate transport capacity, but either really decrease chloride or really increase chloride. And so it's quite confusing, and we still are quite confused with this work and trying to understand it. But our current kind of hypothesis is that it just seems to be a disruption of intracellular chloride homeostasis, particularly in glial cells, that is linked to this episodic ataxia phenotype. And we're now looking at a bunch of other mutations. One was we were actually approached by a neurologist in New York that had identified a family with episodic ataxia, and they have a mutation in EAT1, and we haven't done the Drosophila work yet because they're still generating the fly. So this is a little bit misleading, but just to show you that it's actually another one where we see increased chloride, totally fine transport capacity. So this will be another interesting one to see. If it does really sit here. We know its reverser potential sits here, but what's it going to do in the larvae? And so we're working on that at the moment. Okay, so in summary, what I've shown you today is a structure of the chloride conducting state of the glutamate transporters. This structure really explains how you can have one protein that can both do coupled transport in this closed off kind of transport domain, but also allowed this uncoupled chloride movement through the interface. And I've also talked to you about this disease, episodic ataxia type 6, which is linked to mutations in the gene that encodes human EAT1. And using a combination of experiments, we have identified that, whether you have increased or decreased chloride conductance, you seem to have an impairment that may be linked to the disease. And I also talked to you about this M to R mutation, which is a specific one that seems particularly unhappy. So as I mentioned earlier, we're really interested now in trying to find some functionally selective inhibitors, because these will be useful not only to uncover the role of these two components of the glutamate transporters physiologically, but may also be able to lead to the development of inhibitors that, for example, could prevent this large chloride flux, but still allow glutamate transport to occur. And that's about it. So the most important slide is acknowledgements. I already talked you through the main contributors to the work from my group, Quinny, Isha, Pep and Rosie, our collaborators at McGill, Nick, who we're now collaborating with at New York University, who's letting us know about some different mutations. The MD work was really conducted by Shashank Pant in Ahmad Tashorshid's lab, and of course, Alistair and Megna, who we collaborated with at UNSW, where we use the Artica that is funded, that is part of the Victor Chang Innovation Centre. Also, we're now using microscopes at Sydney University and also down at Monash. And of course, now I am an affiliate member of CEMMP, and like to thank the synchrotron as well for some of the earlier crystallography work, funding from NHMRC and ARC, and all of the transporter biology group. Members are both old and current. Thank you.
Thank you very much, Renae. That was a brilliant talk, very informative. I'd like to open for questions from the audience. If anyone had any, just raise a hand or type in chat and I can ask the question as well if your microphone's not working. Joe, go ahead.
Hey, Renae.
Hey, Joe.
Thanks
how
for
are
the
you?
amazing talk. Great. I was just wondering, because you said after it completes the import of glutamate and the sodiums, then it transports that potassium back the other way, and I guess that powers back to the outward facing state. I was just wondering, through your MD simulations or other data, if you knew if the chloride conductance occurs during that return to the outward state, and if there's any difference in the amount of chloride that gets transported as opposed to the conformational chain inwards as opposed to outwards. Does that make sense?
Yes, it does. And yeah, I must be sure it said that. It appears to be exactly the same. So it is activated, and we've done studies actually understanding how the potassium coupling works. We've nearly got it, but it's been very hard to sort that out, mostly because we now know from work from Olga and some of our functional data we haven't published yet, it's really overlaps with the sodium and glutamate site. So it's always been really hard to tease it out with mutagenesis, but we have used that reverse transport activated chloride conductance, we call it. So the chloride conductance activated as the transport domain coming back to investigate some of those mutations. So it's definitely there. The properties seem to be the same. It's the same selectivity sequence. It seems to be, the properties are the same. It may be open for slightly less time, not exactly sure, but it is thought that that's quite a slow part of the step, the whole process. So the binding of glutamate and that initial movement in is quite fast, but the release and binding of potassium and coming back is much slower. And these transporters are quite slow around 100 millisecond turnover when you think about receptors and other processes that are going on around them. But yeah, that's a good question. It is activated. It seems to be the same, have the same properties, but it's a bit trickier to study and hasn't been studied anywhere near as much in depth.
Oh, cool. Well, thanks again for a great talk.
That's all right. Thanks.
Did we have any other questions from the audience?
Sorry, I may have spoken too quickly. I always find it hard to do these talks when I can't see anyone. You're just like in a bubble. Hi, Emily.
Hello. Thanks for a great talk, Renae. Do you have a question? So with this M128R mutation, obviously it's really detrimental to the protein function. How are patients surviving, I guess, with this?
Yeah, so I mean, the patient is heterozygous, so I think that's part of the reason, whereas our flies are homozygous. But interestingly, I think what it shows is that these transporters are most probably not as important as we maybe thought they were. I think there are other transporters, so EAT2 is very highly expressed on glial cells as well. EAT2 is, I think, meant to make up 1% of brain protein. It's a really highly expressed protein. So E1 is important, But I think there's other transporters around that can compensate.
Yeah.
And I also think even when we see the differences in the chloride properties, it has this episodic nature, because I think there are obviously other chloride transporting proteins, like, you know, the sodium potassium chloride co-transporters and other mechanisms that will come in to regulate chloride.
Yeah.
But it could be that these are periodic because they're activated by a glutamate release, by a synaptic event. You get dysregulated chloride, but then it somehow recovers. So I think it's both the episodic nature, but also the fact that there's other transporters that clearly can cover for EAT1. But yeah, it is quite surprising that they would have fairly disrupted EAT1 function and seem to be okay.
Yeah.
Well, they have the episodic ataxia, but yes. Thanks.
I might ask a question if there's no one else who wants to jump ahead. So I was just curious about the electrophysiology studies that you were doing. It's not really a specific question, but if you could provide some more detail on the sort of conditions you were trying to induce upon in your experiments.
So I didn't really explain that well, I suppose. So we use LSATs from Xenophus levus frogs and use 2 electrode voltage clamp electrophysiology. So it's a bit different to patch clamping, where we have 2 electrodes, one clamps the cell and the other one measures the current required. So it allows us to look at glutamate activated conductances, but we can change, obviously, the conditions of the buffer that the cells are sitting in. We can manipulate the anions, we can manipulate the cations. We can, you know, manipulate the substrate conditions and inhibitors and all the rest to be able to, I suppose, tease out the different components. So the electrophysiology is a really nice tool. You can't use it for all transporters. They've obviously got to be electrogenic, but it's really a very nice tool for us to be able to look at the contribution of the 2 components. And by comparing that to radiolabeled glutamate uptake, which just measures the glutamate uptake alone, you can get a good handle on what parts or what, you know, components of the process you're impacting by making mutations or by varying conditions. So they're quite complicated little proteins, and they have lots of different conductances that can be studied in different ways, but electrophysiology definitely allows us to do some pretty neat experiments with them.
Yeah, definitely. I think it's interesting how you've been able to manipulate it with all these different ions in play, for sure. And kind of a broad, hard to answer question, I guess. The development of this chloride channel, it is quite interesting. You mentioned that there are many other chloride transporters that function within these neurons. Do you have any idea why linking a chloride sort of transient channel to glutamate reuptake would have developed? If that makes sense as a question.
No, definitely. And I think, I mean, I don't really know. I think some of these archaeal ones and bacterial ones in particular are very slow transporters, we think. And they could actually be more like amino acid gated chloride channels. So they might be sensing amino acids. I mean, GL2PH is from an organism that was found in volcanic vents very deep in the Sea of Japan. So high temperature, high pressure. They're these extremophiles. They live in very harsh conditions, very high salt concentration, but very low amino acid concentrations in the sea, obviously. So it's been proposed that they might be more of an amino acid sensing mechanism. You know, how that then translates to physiology of the human brain or, you know, higher order animals. Really the best proposal, and I'd say it's still a proposal, is this idea that because they're electrogenic transporters and you have net transfer of 2 positive charges per cycle, because glial cells are virtually covered in a sea of these proteins, even if they all just have 1 turnover, you'll very quickly depolarize the membrane. So you will have an effect on the membrane potential. That will then slow down further rounds of clearance. And so being able to have negative ions move in to counterbalance that positive charge could be a way that the transporters can function over a wider range of conditions. I think that's the best way to explain it. it's always a little difficult because we're looking at studying them in oocytes, which is obviously not in a synapse, and it's a bit different, and the concentrations of proteins are different, and we have flow of solution over our cells. So it is different to what would be happening within an enclosed synapse. But that's the best option I've heard of, is that it might just help those transporters have another couple of turnovers, which is enough to clear the glutamate and get it down to those low concentrations.
Great, thank you. If there were no other questions, then I'd like to thank you very much for that talk, and I'm excited to see what new answers arise with further research. I'll just like to thank the audience as well. Oh, Sarah, is that a question from you, or is that?
I think it's a clap. I think it's a clap from
Sorry,
Sarah.
thank you.
Yeah.
I was just clapping. Sorry, great talk, Renae.
Thank you.
No worries.
Yeah, sorry, it was a bit electrophysics heavy for this crowd, but hopefully you found it interesting.
Yeah, I'd just like to remind the audience that the next seminar will be on the 11th of July, and thank you for attending. And thank you again, Renae, from myself and on behalf of the centre as well, for the great talk.
Thank you. See you all.